Welcome to Jaan Aru from Estonia at the University of Tartu. So in this semester, we decided to have our CCNB seminar under the topic consciousness and perception. So we were looking for speakers who were doing outstanding work, particularly in the field of consciousness research and the neuronal mechanisms that contribute to consciousness or that allow consciousness. And there in the last years, multiple of us has seen several papers by Jaan Aru. And so we are very happy that he's going to open this lecture series for us. So Jan was actually, he's having like quite some links to Berlin. So he was studying at the Humboldt University psychology, psychology, but as a diploma. And then after this moved to Frankfurt and doing his PhD at the Max Planck Institute for Brain Research. And then later on also doing a postdoc with Matthew Larcum, working on the cellular mechanisms that contribute to consciousness, something that we are very curious to hear about, particularly today in the presentation. And now he is, after being in between senior researcher, he is now associate professor at the University of Tartu at the Institute for Computer Science. So we're very happy to have you. Thanks for doing this, Jan, and I'll hand over to you.
Thank you very much. And I'm, of course, super happy to be here with you today. As you already heard, I have strong links to Berlin, always happy to be there, even virtually. And so this very strange place called University of Tartu happens to be in my home country, Estonia. So that's why I work in that very strange place. I'm an Estonian and I really like it here. But actually this work that I'm going to tell you about was also done in Berlin, in the Humboldt University. And I will talk about that in a few moments. But maybe the link goes back even further. As Timo said, I started my studies, my undergrad in Humboldt University, and there 17 years ago, I fell in love with the problem of consciousness. And ever since then, I've been really curious about of how the field moves and what is happening. And currently, we are in a very interesting time in consciousness research, because there are people like me who would say that we really don't know and don't understand how consciousness comes about. And then there are people who are pretty sure that they know how consciousness comes about, and they are, for example, the proponents of these four very prominent theories of consciousness. So I won't really go to explain all of these theories. I guess you know a bit about them. So IIT is the integrated information theory. Then we have the global neural workspace theory. Then the higher order theory. I should have maybe added the name Hakwan Lau here, who is one of the authors in the later stages. And then the recurrent processing theory of Victor Lamme. And these four theories, they are really very prominent in the science of consciousness. So people study them, and now people have taken a really important approach to test these theories and pit these theories against each other. And maybe you have heard about it, maybe not, but the Templeton Foundation in US has given roughly 10 million euros to test these theories against each other. And for consciousness science, that is a lot of money. And of course, one could say that, yeah, this makes sense to actually test to, for example, see where the IIT and global neural workspace theory make different predictions, and then to have this adversarial comparison. It makes sense. And I agree, it is a great project. The labs doing it are great. The people running it are also amazing. However, when I first heard about it, that 10 million euro is going to basically into testing these theories, then I thought, well, is this really the best way to basically spend money in consciousness research at the present time? And I would like to convince you, or I mean, not convince you, but at least show you this alternative view that, actually, I think times are good for developing new theories of consciousness. And I will try to tell you about one of these theories, but let me first just say why I think the time is good and that we actually need to develop new theories of consciousness. One of the main reasons is that if you now look again at these four theories, then you see that they all have been founded more than 20 years ago. And of course, age by itself is not the problem. Some things get better with age, like some wines, right? However, the problem here is that over these 2 decades, many things have happened in neuroscience. And I would like to tell you about 2 of these revolutions. And the first, of course, is the revolution in the methods of neuroscience. So 20 years ago, we did not have optogenetics. We did not have any of these fancy tools that we today use to actually control the brain and dissect the circuitry. So here's just one slide. I won't really go into that, but here you see that basically we can today genetically identify cell types, we can activate them, we can deactivate them, we can block connections, we can do all kinds of stuff that we could not do 20 years ago or 15 years ago. So there has been a revolution, and one example of this revolution is that all of these theories were still very much based on the idea that a neuron is something like an integrate and fire point neuron, meaning that a neuron gets inputs, and then if these inputs cross some kind of a threshold, the neuron fires. However, now through to the optogenetic revolution, we actually know that this is how a computation in a neuron looks like, right? And again, I won't go into all of these details, but the point of this image is just to illustrate that things are rather complex, and they cannot be captured by a point neuron. So I will come back to this. I will tell you more about the complexity of the dendrites, but here for now, just let's take this point that a lot has happened in neuroscience, but not only in neuroscience, right? The other revolution I would like to mention, and that is important for me personally, is the revolution that has taken place in our understanding of the effects of top-down processes. And if you look at these examples now here, that you all know, examples of how the top-down effects affect conscious perception, then some of them are at least rather old, right? And they are known. They were known 20 years ago also. But all these effects were rather marginal. So if you look into these papers of these theories, then these contextual effects and top-down effects were not really so important. One could probably say that for Lamme's theory of recurrent processing, they were more prominent, but if you go and read his papers, then it's not that he puts a lot of time into explaining this and saying that, look, this is the fundamental aspect of consciousness. However, today, in part because of the prominence of the predictive coding ideas, the idea that conscious experience is shaped by top-down processes is fundamental. It's basically everybody agrees that there is this effect of top-down processing, and these effects are all well known. But I just for illustration, I wanted to show something we're working in my lab on. So here you have an image that is it's like a constellation. right? So here is a real object embedded in noise, and your task is to find what kind of an object here is. Some of you might find it, others not. But now, when I tell you that look for the classes, then many of those who previously didn't see it will see it now. And the question is, what happened in your brain? So if I remove this word, you still probably will see the classes, right? But what happened? What changed in your brain? And can theories of consciousness explain that? How this top-down knowledge changed on one shot our conscious perception. So this is a question that is very, very important for me. How can so this is just a slide to show how we have generated hundreds of such stimuli that we can then use to study different things. I won't go into that now, but the point I want to make is that what will these theories of consciousness say? How can they explain these kinds of effects? And of course, we could take any of these theories and make extra assumptions. We could add some extra details so that these theories can explain them. Of course, they can. But the question is, should we really try to patch together these old theories of consciousness? Or should we rather say, look, we know now much more about the brain. We know now much more about some other stuff, and we should actually be developing new theories of consciousness. So I'm not saying that, we should not test these old theories at all, but I'm just saying that, we have to ask what is the right balance between testing old theories and developing new ones. And I feel that this balance has tilted a bit, especially with this major funding from the Templeton World Foundation, that this balance has tilted a lot towards testing old theories. I apologize. And as you can already kind of expect, I propose that, instead of that, we should also consider new theories of consciousness. And I want to present to you one of these theories that we presented last year. However, very importantly, my point is not here that this is a much better theory than the others and that this theory is the best theory. No. My point is that this is an example of a theory that has taken these things into account. So rather than saying that this is a perfect theory that will should kind of make everyone happy, I'm saying, look, this is one particular theory. This is one example. And I encourage all the young researchers to actually go and think about how else they could construct a theory. So my point is, instead of, spending all of our research time on these old theories of consciousness, we could admit that we don't know, and we could develop these new theories. So I will tell you now more about this theory, but let me first grab a sip of drink. So if anyone has a question at this moment, then please use that chance to ask. Yeah, and sorry for that. I had another talk today, so it's just too much for my voice system. Okay, so what is this theory about? The central players of this theory are the layer 5 pyramidal cells. So they are in your cortex. The cell bodies are in layer 5. However, the dendrites of these cells arise and reach the really top of the cortex, layer 1. If you consider that in your brain, these dendrites are sometimes millimeters long, several millimeters long. So they are really huge dendrites. And why these cells are important? So here's a nice illustration. So you have the cortex here, but then you have subcortical structures. And there are some cell types, like the layer 6 cells, that do these thalamocortical loops. However, layer 5 cells are much more powerful. They send information to the striatum, to the non-specific thalamic nuclei, and they even send projections to the brainstem. So fundamentally, layer 5 cells, or a subclass of them, is the most important output of the cortex. So your cortex can do all kinds of fancy computations, but if you would abolish all of these layer 5 cells, it wouldn't mean nothing. It wouldn't do nothing to the system, because layer 5 cells are the main integrators and output of the system. So if you look at these cells, then I told you already that they have these amazing long dendrites. And then in our work, we really have taken this kind of stance that we could here say that we have at least 2 compartments. So one compartment is around the cell bodies. It's the so-called basal compartment. And the other compartment is then around layer 1, layer 2, so the top compartment, which we call the apical compartment. And yeah, one thing is to just say that you look, here are 2 different compartments, but it was Matthew Lacum, who's now in Berlin, in Humboldt University and Charité, who said that, look, it's not just only 2 compartments, but they receive fundamentally different input. The apical compartment receives top-down information, or he likes to call it the context, whereas the basal compartment receives bottom-up, or he calls it feature information. So this is, I think, pretty exciting. This idea is exciting, that, you know, there are cells in our brain that have distinct compartments that receive functionally different input. And I mean, nowadays there are many studies that kind of support this idea. Let me just put here one review for you that shows that, you know, in layer 1, where the apical dendrites are, you have projections from higher order cortical areas. For example, this A1 is auditory area, right? You have projections from perirhinal cortex around hippocampus. And then you have projections from different types of thalamic nuclei. All of them project to this layer 1. Some of them also project to layer 5, but this kind of thing that, you know, there's this projection going to layer 1 and this does something specific is well established in neuroscience. So Matthew Lacum did not only kind of propose this idea, but he did exciting science that's published in Nature now more than 20 years ago. He did these amazing experiments where he put not only 1 electrode inside a cell, but actually 3 electrodes. And then what he could do is he can give a bit of electrical current into these different spots. So here we have the apical compartment, here the basal compartment, and then he can do all kinds of combinations and check what happens. And I won't explain all of that, but the point is here on panel D that if you combine the apical and basal activation simultaneously, then something else happens. You will actually be able to see that there's a burst of activity with a strong signal of calcium currents. And in short, there is basically a mechanism within each cell that says to the rest of the brain, look, here is a match between these two streams, between the bottom-up input and between the top-down input. And so you can understand why this was a Nature paper. I think it was a really amazing finding. Find, on the level of a single cell that there is an association mechanism. So if you combine top-down and bottom-up, then you have this burst of activity and the rest of the brain basically knows, oh, here was a match. Something was found here. Okay, so as I told you, Matthew, in his papers, he called these things context and feature, but here's a very important point that we make in our paper also, that, it does not really matter how you call them. So here I have just expanded this zoo of different names, right? That you could say that this is top down, that the apical compartment receives predictions. You could say that they are high order representations. And to the basal compartment, you could say that these are prediction errors, or you could say that this comes from the external world. The point is that the exact name does not matter. What is important is that anatomically, they are distinct inputs, where one comes from within the brain and the other is rather somehow reflecting the data or the external world. So you might call them by different names, and people do. Some people use the phrase top-down, bottom-up. Others are very keen on prediction and prediction error, etc. The point is, biologically, they are different streams going onto this one neuron. At this point, you might be wondering, but what on earth does all of this have to do with consciousness? And until now, it seems that not much, right? However, when I went to Berlin to study with Matthew, then this postdoc, Mototaka Suzuki, showed me a result that I will now share with you. So he did these amazing experiments where he shone light only on the apical compartment. So he optogenetically activated the apical compartment, and then he looked at what happens on the basal compartment. So he measured here with electrodes the neural spiking activity. When the animal was awake or conscious, then you could see this activity propagation from the apical compartment to the basal compartment. But the crucial finding was that under anesthesia, this did not happen. So he goes and activates the apical compartment, but there's nothing, no activity on the basal compartment. So the same activation in the awake conscious state led to activation of the basal compartment. But now under anesthesia, it didn't. So he tried several different anesthetics, I think 4 different anesthetics, and always the same finding. He went to a different brain area, again, the same finding. So this finding was really astonishing. So when I went into the lab of Matthew Lacombe, I didn't actually want to work on consciousness. But when I saw this result, then I had to, because this was simply so unexpected. And here's the point I already tried to make in the beginning, that look, this was published now in 2020 in Cell, and 20 years ago or 15 years ago, we could not have expected this kind of a thing, that within a single neuron, there is a mechanism that changes with the state of consciousness. That basically it looks, when we look at it, as if there's some kind of a gate there during the unconscious anesthetized brain. And in their further research, they went on to show actually that this kind of a gating here, so controlling whether the activity goes through or not, is done by higher order thalamus, which has very specific connections in between these two compartments, the apical and the basal compartment, and which they figured out some very specific mechanisms depending on the metabotropic glutamate receptors. If you are interested in that story, in that part of a story, I encourage you to read our Trends in Cognitive Science paper. I think there we give a very nice, well, it's not good to say very nice to your own paper, but I think it's a nice, again I said the word, sorry. it's a summary that you can understand even if you don't have a stronger background in systems and cellular neuroscience. In other words, their Cell paper might be a bit harder to read, but here we try to give a good overview. So this meant that, on the one hand, we already knew that there's these two compartments of the neuron, of these layer 5 pyramidal cells, that represent something like internal and external, top down, bottom up. But now, thanks to Mototaka's research, we actually knew that this mechanism is disrupted when the animal is unconscious. And for us, this was really a big finding and illumination or revelation, because we realized that, here could be a really fascinating mechanism that could explain how consciousness works. And this mechanism is a cellular mechanism, a very local mechanism. So I will tell you now more about the properties of this mechanism. But the point I wanted to make here is that, you could say that this, although this is a cellular mechanism, it gives a very kind of fundamental idea that during conscious states, you have this coupling between the external, coming to the basal dendrites, and the internal, going to the apical dendrites. And that unconsciousness means that you have a decoupling between the internal and the external parts of information. So, and that this is a fundamental feature of consciousness, that these internal and external are coupled. Okay? And I will tell you a bit more about what this all means. We put it into a theory. We call it dendritic integration theory. I myself am always uncomfortable. I don't want to sound too fancy. So if you don't want to call it a theory, please call it a dendritic integration mechanism. I don't mind. The point is simply that, you have these two different compartments, and for consciousness, they have to be integrated. But let me show you now what this theory can explain and what not. But perhaps here are already some questions that need clarification. If you have something more philosophical or fundamental, leave it to the end. But if you have something that requires clarification, then please go ahead. All right. So what are the properties of this theory? I think one important facet of this theory is that it can really explain the relationship between the state and contents of consciousness. We expand on that a bit on an earlier paper that you can also look up. But the point is that those of you who have studied consciousness, you know that basically there are really two camps, or there have been. One is the camp of studying the states of consciousness. These are the people who study the thalamus, the higher order or non-specific thalamic nuclei, and study how thalamus activates the cortex. And these people usually work on the disorders of consciousness, for example. On the other hand, we have this camp of people studying the contents of consciousness. And these people usually They try to study the cortex. They mainly do neuroimaging of cortical areas and see how the level of how being conscious of a particular object changes the activity. But here now we say that, look, these two are coupled. They are fundamentally importantly coupled because the layer 5 cells, the layer 5 pyramidal cells, as I told you, they are the key output to these non-specific thalamic nuclei. They receive input from these non-specific thalamic nuclei. But on the other hand, they also are a key player in the cortical loops. They integrate information from the cortex. They integrate this. So when you're conscious, then these cells allow this activity to propagate. So they allow this activity to propagate to thalamus and within cortex. However, when you have this, when you are in an unconscious state, then this gate is blocked. As I showed you, these pyramidal cells do not propagate information. So you can have some local activity in the thalamus, you can have some local activity in the cortex, but these two, they don't talk to each other because the main communication channel, these layer 5 pyramidal cells, are basically cut off here in the middle, functionally. So this is an important point, and now we say that, look, this explains fundamentally why it is like that when you are conscious, then most likely you are also conscious of some contents. Only in some very specific conditions, perhaps you can be conscious of nothing, but then you could say that this nothing itself is a content, right? But mostly when we are conscious, we're conscious of a content. And when you're unconscious, then you cannot be conscious of a content while being unconscious. These things are intertwined. The state and contents of consciousness are intertwined because anatomically, layer 5 cells sit in the middle of that and shape this communication. So this is, I think, a nice property of this theory that many other theories, they really don't pay attention to this at all. Now, the other point that is very important for me is that, I showed you that this mechanism is very local. So this mechanism is basically cutting the communication between the two compartments of the pyramidal cell. So it's a cellular mechanism, happens within one cell. However, as you can imagine, you have millions of such cells in your brain. And this means that, although it is a local mechanism, then it can have a global influence. And hence, we are very certain that this local mechanism can explain the fact that during conscious states, you have this global thalamocortical reverberation, whereas in unconscious states, not. So I already explained this to you, right? And showed this. But now the point is that locally controlling these cells will control the global activation patterns in the brain. So I very much like this paper by Athena Demirci and others, who basically in the abstract, they managed to say that consciousness rests on the brain's ability to sustain rich brain dynamics. And here, based on our theory, I would add to that, these rich brain dynamics actually depend on this dendritic integration between the two streams. So, and this is how I would explain it, that, if you're unconscious, then you can have local activation. You can have the activation of the basal compartments. You can even have the activation of the thalamus. Perhaps you can even have the activation. In fact, we know that you can have the activation of the apical compartments, but as here you have this gate shut off, then nothing happens. You don't have a loop that can self-reinforce itself, right? This communication is cut off, so you can do local activation. And for example, we think that during non-REM sleep, this is a mechanism of learning, that you actually project the information to the apical compartment, but you do not activate the basal compartment. Whereas in conscious states, you can then build these recurrent loops between the cortex and the thalamus. And these recurrent loops then can, for example, maintain working memory. And as some of you know, in mice, it has been indeed shown that working memory is maintained by this loop of a higher order thalamus through cortex through higher order thalamus. And there are mechanisms that gate this loop and switch it off. One of these mechanisms is this mechanism local to the pyramidal cells. And then, so we think that we can explain with this local mechanism, these effects that have been measured on the global level. For example, here, I want to show this work of Marcello Massimini, basically showed that if you put a TMS pulse to the brain, then in wakefulness, you have these complex activity patterns. Whereas in non-REM sleep, you create only a local activation. And then based on our theory, we can say that, look, in non-REM sleep, you have these gates within the pyramidal cells closed, and you can do local activation. You can locally activate the thalamus, but this will stay local. Okay. What I wanted to discuss a bit further is also that, so far, as you have seen the theory, you could say, oh, it's a theory of state of consciousness. But it's important to say that it's not. It's also a theory of what really happens within a state of consciousness, and we want to explain also the contents of consciousness. So you could think of this like that this top-down information, attention, feedback, context, prior knowledge, it can activate the apical compartment, and by pre-activating it, it can actually lead to a faint stimulus becoming conscious. So if you don't have this pre-activation, then you can have a stimulus that activates the basal compartment, but does not lead to much further processing. This would be an unconscious stimulus within the conscious state. However, if you have this pre-activation, then and only then you can build these strong recurrent loops and be conscious of that stimulus. And here I just wanted to highlight that this is not just an idea or a hypothesis, but this actually has been shown empirically in this very nice paper, again from the lab of Matthew Lacum, where they did something that some people in your institute have done, where you do a manipulation of consciousness such that you manipulate the stimulus intensity. But they did it in mice. Can you imagine how difficult that is? They trained it for several weeks to do these experiments, but then what they can show is they can basically have the psychophysical curves, and then they can look at activation of the brain. And here they were interested then in the activation of these apical dendrites. And here, if you see my cursor, you see that here's a dendrite that really scales well with the stimulus intensity. So if you make the stimulus more intense, the dendrite starts being more active. However, it's a science paper because what they did is they also manipulated this thing. So let's first look here. I hope you can see my cursor. So panel D is a psychometric curve, right? Where you have on the y-axis the detection probability. So whether the mouse reported to have perceived the whisker stimulation or not. And then on the x-axis, you have stimulus intensity. And then what they here did, they did one type of an optogenetic manipulation where they inhibited these layer 5 pyramidal cells, the dendrites, and you can see that the detection probability goes down. Now, and here you have this, you know, at the threshold, at the threshold where the animal is sometimes 50% reporting to see the stimulus, 50% not, then you see that doing this manipulation, so inhibiting the apical compartment, led to the animal not perceiving the stimulus. So this is the, you know, the cool things you can do with optogenetics. And then, as you can imagine, in the following experiments, they did some more optogenetic manipulations. And let me just talk about this last one, where they activated the dendrites of the pyramidal cells. And here you can see, let's go here to the last panel, bottom right, where you can see that, you know, the animal is at threshold, but you can now increase the probability of the animal reporting that, oh yes, there was a whisker stimulation, simply by optogenetically activating the dendrites. And in fact, if you look at panel H left side, then if the stimulus intensity is 0, so there's no stimulus, then if you do this optogenetic manipulation, you can make the mouse report that, hey, I saw a stimulus, or in this case, I whisked a stimulus, right? So this kind of manipulation of the apical compartment shows that you can actually have an effect on conscious contents within the state of consciousness by controlling these apical compartments. Okay, so I see that the time is already running late, but let me maybe go at least to this last property and then maybe make a bit briefer, because as I said, this kind of an effect that this cellular mechanism can explain why top-down effects are so pervasive and why top-down effects shape conscious perception. It's really important for me. So I think, I mean, you remember this figure, but what it essentially says, in other words, is that conscious processing is processing that is inherently affected by these top-down processes, because there is this coupling. In unconscious states, you cannot have this effect from the apical compartment to the basal compartment. However, in conscious states, you have it. So all conscious processing is affected by these top-down processes. And this is, for me, an important point, that, you know, we don't make any extra assumptions. Rather, we really inherently say that conscious perception is affected by the top-down processes. So it is inherent to the theory. We don't need to make any extra assumptions. It is the theory. And then if you go to our example of this, you know, you have this input, you try to figure out what is here. You might be activating many basal compartments, but once I tell you, you look here at classes, then this explains away the activation and activates the right pyramidal cells that now can actually tell you that, look, here is here are classes. Okay, so let me more or less skip this, that, you know, we think that this theory very nicely and again inherently can explain dreams and hallucinations, which again other theories have a more difficulty to explain. They can do it with extra assumptions, but for us this is natural. So if you want, you can look into this paper I also published last year about dreaming. But fundamentally, maybe very briefly, I won't explain these results. The point is that if you activate this top-down compartment very strongly, it can drive perception. And essentially, this was what I showed you when I some minutes ago explained this result by Naoya Takahashi and others, that look, if you activate the apical compartments, you can actually make the mouse report that it perceived this whisker stimulus, although there was actually nothing there. And in our research, which I didn't show you, we have also shown that, you can do this in healthy humans too, that, I mean, not the apical activation, but that they perceive something that is only generated by their expectation. Okay. And let me finish off. So in the end, I have presented you our view of how, we could take into account the modern findings in neuroscience and how we could build a theory around them. But of course, we also acknowledge in the paper that, you could take this mechanism and integrate it with these existing theories of consciousness. So here I made a slide about IIT and global neural workspace theory, but it would also fit with the higher order theory, or of course with the recurrent processing theory. But essentially, as I told you, most of these theories, they take into account, or they basically assume that these are point neurons. But you could simply say that, look, each of these units has these two compartments, and it has this mechanism of being able to switch off this communication between these two compartments. And as we show in our paper, if you would take this into account, then I think both of these theories, or any of these theories, could also win something. They could explain some phenomena that would otherwise be perhaps hard to explain. So fundamentally, again, I'm not against these old theories. I'm not saying that we should throw them all away. I'm just saying that, look, we can have these old theories of consciousness, but we could also develop new theories that take this kind of new findings from neuroscience and also from cognitive science into account. That these are great times for us consciousness researchers, and we don't have to settle for these old theories. We can actually study and develop new theories. So if my talk doesn't do anything else, I hope it encourages young researchers amongst you to kind of think for their own and not put their whole career into studying someone else's theory. And perhaps the last thing is to say that, of course, whenever I present to people like you, then the question is, hey, is this relevant for me? Because how can I actually study this kind of a thing? It's a cellular mechanism, very nice, but what does it tell for me? And then I want to just mention that we are thinking about also trying to do this with fMRI. I personally do not have much experience with that, but I have at least one potential postdoc who is very excited to try this out, to try to do a model-based fMRI based on this theory. Then in the 70 fMRI that can resolve layers. So here I just want to throw this out, that if somebody wants to give some tips or hints or comment on this idea, then it's very much appreciated. But for now, I want to leave you with this idea that, these are amazing times for consciousness research. There are new facts and findings coming basically every week. So let's not only settle for the old theories of consciousness, but let's have fun and develop some new theories. Thank you very much for the patience. I'm happy to take some questions.